what we're going to be doing today is finishing up our blood vessel chapter and then spending most of the time just reviewing for the lecture exam itself. So going over questions that you may have from the exam objectives or topics that we covered, including blood chapter, heart chapter, blood vessel, and hemodynamics chapter, right? Before we do that, I had promised to kind of review the structure of the exam itself, the anatomy of the exam itself. So remember that for the lecture quiz will be due tomorrow night by midnight. So the lecture quiz is always due 2 days before the lecture exam. And that way you can kind of get the results from that, right? So your results from that will be available immediately. You can go back to the question and kind of see as an indicator where things went smoothly, where you may need to apply a little bit more of your study time in the remaining 2 days leading up to the exam, okay? So the exam itself, and the quiz will require Respondus Lockdown Browser, not because it's closed book, but just because we want to make sure that Respondus Lockdown Browser is still installed in your device okay, it's still working okay. It's going to give you one more time to practice with the environment check to make sure that you're checking everything around you, that you have the camera repositioned to your face again afterwards, etc. Okay, so then when we get to the lecture exam, due 11:59 p.m. on Saturday. You can do it anytime on Saturday, right? So if you have to work that night, you could take it earlier in the day and get it out of the way, whatever. That one is mostly multiple choice. The quiz was all multiple choice. The exam is mostly multiple choice. So it's like 40 multiple choice questions, probably. Each one worth 2 points. So that means 80 points on average, I think. 80 points or 80% of the exam is going to be multiple choice questions. Equally divided to a certain extent between our 3 chapters. So about a third of the questions divided equally to each of the chapters. And then we'll have 5 vocabulary questions. Each of the vocabulary questions is only worth 1 point. But in most cases, the way I have that designed is there's the word, and you're typing in a definition for it. Okay? So we know we have our vocabulary list as well as vocabulary flashcards available electronically in Canvas. So make sure that you're practicing with those so that you have an adequate definition in mind for if you get asked, when you get asked, some of those questions on vocabulary. It will only come from that list. Okay? In addition then, so that's 5 points. The remaining 15 points comes from your short answer questions. So there'll be 3 different short answer questions that will come from the exam objectives that we see in bold, right? From blood, from heart, from blood vessels, and hemodynamics. So they'll just kind of be randomly assigned to you. I don't know which one you're going to get either, but it'll be from that list. So make sure that you're reviewing your exam objectives, and particularly make sure that you have written up and practiced with some good answers for those that are in bold, because those will be your possible short answer questions. And those are just, free answering. You're typing into the open space as to comparing, contrasting different types of blood cells, or the different blood vessel types, or doing a tracing of blood through the heart or something like that, or talking about bulk flow and the different pressures involved that get fluid to movement across capillary walls, or describing what's going on during an EKG. What are the 3 active waves, and kind of what's going on during each, things like that. Okay? So with those, with a multiple choice, just like on the quiz, those will automatically grade. So when you get done, you'll get a partial grade. Don't panic. It's a partial grade based on just the multiple choice that our Canvas is able to quickly assess for you, right? But then next week, much like when we do lab quizzes, right, the following week is when I have a chance to get in there and grade the other components, grade the vocabulary section and the short answer section. So the grade that you see immediately will be just based on the vocabulary out of 80. So then there's a possibility of an additional 20 points coming your way based on how you performed in the vocabulary and the short answer section, okay? And then as I get to those, of course, it'll update your grade and you'll see a bump, right? or not expecting that would not change. So make sure that you study up for all of the different sections of the exam, and then use those resources, the vocabulary list and those bold exam objectives, to prepare for those last two sections. Those should be the easiest sections because it's very, very guided as to what you're expecting to show up in those sections, right? It still takes preparation, still takes practice, so you have a good answer, but at least it's like, I don't know what it's going to be. I do know what it's going to be. It's going to be in those lists that we have, okay? And won't happen to you guys, but always make sure that you put an answer in there, right? Even if you're unsure, you're like, gosh, that vocabulary term, I don't remember that one. Put something in there, right? It's not like an SAT where you're going to get negative points or something like that. I'm all about partial credit. So if you are putting in an answer and there's certain parts that are correct, you're going to get points for all the parts that you had correct. If you're missing some things or if you say some wrong things, obviously you're going to lose some points for that. But partial credit is a great aspect. It's one of the reasons I put those sections in there, right? Multiple choice questions, even if you narrowed it down to 2 out of the 4 answers and then you went the wrong direction, you get no points, even though you were able to successfully get rid of some of those answers, right? At least with these other ones, you have the probability of partial credit. So make sure that you put in an answer for everything. Keep an eye on your time, because these are timed. It's like 75 minutes, like a typical class period. So you should have definitely adequate time to go through all the sections, so you don't need to hurry. I think you can access all sections of the exam in the order that you like. So if you want to move forward to the short answer questions, take a look at what those are, answer one of those, and then go back and forth. It's entirely fine. But just make sure you've answered everything, right? It gives you that little window in the upper right-hand corner. It shows you a check for everything that you've answered. Just make sure all of that is covered. Scroll through, double check things before you submit. Okay? All right, so questions about test format or anything like that? Yeah?
I'm sorry, I missed this part. So vocab, do we write out the definition?
Mm-hmm. Yeah, so I'll give you the word and you provide the definition for it. So typing it in there.
Scratch paper, right?
Good question. Yeah. So if you would like to have scratch paper to be able to, like, you know, come up with, you feel like you want to get some of these things down, like you would normally write in the margin of the paper or something like that. If you had an exam paper, that's totally fine to do. When you're doing your environment check and you're resetting your camera and stuff like that, if you have a blank piece of paper there, right, it's going to show up in your environment check. Just when you set up the camera again, say, oh, here's my scratch paper. Show me that there's nothing on it, right? And that's totally fine. Then it's totally okay for you to be like writing things on it and, you know, doing graphs if you want to and stuff like that, to, especially for like, I don't know, ECG or, you know, the action potential, stuff like that. It may be helpful to kind of draw it out and then be typing in your answer when you're looking at it and describing it. Okay? Okay. Make sure nobody else is around. Make sure it's a quiet environment and stuff like that. Everybody else in the household should know that you are unavailable at that point, right? And then, you know, once you set up everything at the beginning, just, you know, the camera will be running and recording, but just don't worry about it. Just start reading it and do it, right? So unless you have your scratch paper there, right? Scratch paper, you'll be looking at that and doing that. But other than that, we're expecting you to be looking at the main camera, looking at the screen and stuff like that. So every once in a while, if you're like, oh, you're trying to think of something, that's okay. But we don't want to see a whole bunch of, like, I'm looking at the video like, what's over there? What? No, just focused on this, right? So in any case, it should be all set up. So just ignore everything else and focus on getting the test done, submit it, and then the next week you'll have results in terms of a full grading of it. Okay? If something happens unexpectedly, tell the camera and then send me a follow-up email when you're done. So we're expecting that you don't have to go to the bathroom, right? Go to the bathroom beforehand. So we're expecting that you'll be there the whole time and nothing's going to happen, but sometimes things happen, whatever. Storm, lost power, or something like that, or dog threw up or something like that, right? Kick it to the side for a while or whatever. Okay? Or if a kid comes in and throws up, whatever. Sometimes those things happen. I've seen it. I had this one student, extremely dedicated student, went up and beyond. I did not expect this. She was not feeling well, decided to go ahead with the exam, was actually throwing up during the exam. Like, she would just be like, working, they'd be like, ah, and then she'd like working, ah, over there, and then keep working, right? Something happens like that, again, just let me know. It's okay. We can work it out. Okay? Okay. So we just had a few additional slides of the blood vessel chapter that I wanted to finish up. I wanted to touch on our bulk flow, our fluid exchange at the capillaries as well, because that was the last major topic that we hit. We'll finish up those other few slides, and then we'll just open it up for review, questions that we want to go through, topics that we want to discuss, and things like that. Okay? So, this, as we're talking about, is also known and sometimes phrased as bulk flow, because we're looking at the flow of water between the capillaries and the tissues, and sometimes, right, sometimes various things in the fluid will transport with it, kind of like if you're pouring lemonade or something like that, you get ice cubes and stuff as well as the lemonade itself. But this is how we do an exchange at the tissue level. Now, for other solutes, like oxygen, carbon dioxide, nutrients, waste products, and things like that, the main driving force behind that exchange is going to be basically diffusion, right? High concentration, low concentration, it's going to be moving from high to low in whatever direction. It may be facilitated if it needs a transport molecule or something like that, but it's basically diffusion. This one operates differently. The forces behind moving water is going to be different. It comes down to two forces, okay? Those main forces are going to be, right, hydrostatic pressure. This HP that we see here with the C, that is hydrostatic pressure in the capillary itself. So hydrostatic pressure, which is the same as blood pressure. So we can call that blood hydrostatic pressure. So that is the main force that is pushing fluid out of the capillary at the beginning of it, right? Because we have a relatively high hydrostatic pressure inside of the capillary at the beginning of it, that overcomes this blood osmotic pressure that we have inside of the capillary, trying to draw fluid back in. So we have those opposing forces, and at the beginning of the capillary, the hydrostatic pressure is the one winning between those two, right? So this number is higher than this number. So when we add them together, we have a net filtration pressure of 10 millimeters of mercury pushing fluid out. Now, you don't need to know numbers, but we do need to know at the beginning of the capillary, blood hydrostatic pressure is higher than the blood osmotic pressure, and so fluid leaves the capillary. That's the important thing, okay? By the end of the capillary, because that hydrostatic pressure is reduced, we know that due to resistance and drag and stuff like that, it's constantly going down. By the end of the capillary, now that hydrostatic pressure is low enough that the osmotic pressure is now higher. And so in that kind of tug of war, now we get a net movement into the capillary because the osmotic pressure is drawing that fluid back in, right? So we still have the same two forces, but now, comparatively, the blood osmotic pressure is stronger and it draws the fluid back in instead. So we have the movement of fluid, of water, out of the capillary at the beginning, which is exactly what we want, delivering to the tissues. But before we leave that capillary, we're drawing most of that fluid back in so that we're not constantly loading the tissues and overloading the tissues with water. Okay? Okay. So the key thing about that, like we said, this is occurring in the capillaries. These are our exchange vessels. So whenever you hear exchange vessels, that's talking about capillaries. All the other ones have the thick walls, all three layers, all three tunics. Capillaries have a single layer. Simple squamous epithelium with a little basement membrane underneath it, right? Super thin that it facilitates the transport of things across it. So this is happening at the capillaries there. We have the two forces involved that ultimately allow fluids to exit the capillary at the beginning and then come back into the capillary and re-enter the blood by the end, before we then go on to venules and on our way. Okay? So, how does that seem in terms of that bulk flow? Any questions about that? Yeah.
So the osmotic pressure is inside the vessel.
Yeah, so it's called blood colloid osmotic pressure. It's due to a number of factors, mainly due to albumin, that one plasma protein, where we have a lot of that inside of the blood, and that creates that osmotic pressure, right? Because it's loaded with solutes, including those plasma proteins, on a relative scale, there's more water outside of the capillary than inside, so the water wants to, through osmosis, go into the capillary. So the presence of those things in the plasma helps to create that draw. It's just the pressure of that is not strong enough to overcome that hydrostatic pressure at the beginning. And so it's always there, trying to draw it back in, but it's overwhelmed with the hydrostatic pressure, the blood pressure at the beginning. So we have this net movement out of the beginning, and once that hydrostatic pressure, that blood pressure weakens a bit, now the tide's going to turn, and now the osmotic pressure is going to be strong enough to suck it back in.
So is that pressure that's coming, it's just from the heart pumping, and every time it pushes a little bit more, it's repeating that cycle over and over through the capillaries. Is that what it is?
So definitely the hydrostatic pressure is the same as blood pressure, like we talk about. And so that is generated by the heart and maintained by the blood vessels, and it's pushing things around the circuit. That hydrostatic pressure does come from the heart. We know that in arteries, we do see this constantly rise and fall, right? We see this pulsing sensation, systolic, diastolic. But by the time we get to arterioles, and that's a good review for the different blood vessels and kind of what they do and why they're there, the arterioles dampen that so that we don't have that pulsing anymore. Now, the pressure, while it's reducing, is not like doing that kind of wavering back and forth and back and forth. So it's still kind of pushing it around like that. But the presence of those solutes in the blood is what's causing that blood osmotic pressure, that kind of constant try to draw of things back, of fluid back, of water. Nadine, did you have a question or no? Okay. Okay. So other things, as we were finishing up, we were talking about maintaining blood pressure and things like that. We talked about, every once in a while, we talked about diseases, disorders when things go wrong. Now, most of the time, like I said, those would just be used as examples and not be on the exam, but there are some that do show up. Like we're talking about hypertension and hypotension. Those are chronic issues of slightly elevated, slightly depressed blood pressure for various reasons. When we're talking about shock, just like an emotional shock, this is more intense, okay? So more of a critical drop in blood pressure all of a sudden, for some reason, is what we're talking about in terms of shock, where there's inadequate blood flow to vital organs, etc. Right? Hypotension is just like the overall pressure of the system is a little bit lower, so things aren't being pushed through quite as readily. In this case, it's like, oh, we're almost cutting off flow to things, and that's going to be a problem. So circulatory shock is that kind of precipitous drop of blood pressure, which can come from a variety of things. Now, as we've been discussing, one of the reasons that I mentioned it is that when we're talking about blood pressure and blood flow, we keep highlighting the fact that it can come from three different things, right? Aspects of the blood affect blood pressure and blood flow. Aspects of the heart affect it. Aspects of the blood vessels affect it, right? So if you have too much blood volume or too little, that affects blood pressure, right? You're packing more fluid into the blood vessels, and that has a pressure and exerts a pressure on the walls, etc. So if you have a drop in blood pressure, that can come from a large-scale drop in blood volume. So if, for whatever reason, like a bear bit your leg off, oh, that's a problem for various reasons. Guess what? You've gone into shock, hypovolemic shock. Hypovolemic shock, your heart and blood vessels, despite their best efforts, they're going to increase heart rate, and we're going to do vasoconstriction and stuff like that. But because a lot of your 5 liters of blood have leaked out onto the forest floor, they're not going to be able to maintain blood pressure, right? So the problem is a low amount of blood volume is leading to that big drop in blood pressure, okay? There's also vascular shock. Vascular shock is a problem with the blood vessels themselves. So we know that they're supposed to be constricting and dilating in order to maintain blood pressure, not too high, not too low, and making sure that it delivers adequate blood to our active tissues. And there, as we were talking about before and we're part of a review today, there are various local factors that can influence whether a blood vessel, like an arterial, constricts or dilates, right? But if your blood vessels are not doing that properly, and if a bunch of them dilate, then that's going to drop your blood pressure. Because constricting increases blood pressure. If too many of them are dilating, now it's going to drop the blood pressure through the floor. One of the examples of vascular shock is if somebody has an extreme allergic reaction. Have you heard about that one? So like anaphylaxis or anaphylactic shock, right? So in that case, what's happening is, so we can use that as an example in terms of how the body would normally respond to things. So if, like we said, you get a mosquito bite, you have a cut, or a bee stings you or something like that, you have a change in local factors that are then affecting blood flow, like vasodilation, right? We're opening up the blood vessels, we're getting more flow there because we have damaged tissue, we're being attacked or something like that. We need to get our forces there, right? So that is in a particular area, but if you have an allergy, oftentimes, again, you're moving blood there to attack whatever it is, right? If you have an infection or if you have an allergy is where there's something that has gotten onto or into your body that your body is treating like it's an infectious agent, like a pathogen. It's treating it like it's a bacterium, a virus, a fungus, something like that, but it's not. It's pet dander, it's peanuts, stuff like that, right? A peanut's not going to get inside of you and grow into a peanut plant, right? But for whatever reason, sometimes our bodies go awry and they treat it as that. And so they're looking to attack it, and sometimes too vigorously. So usually we have local vasodilation in order to get our forces there. But in some cases, the response is so extreme, it's like, I don't know where we're being attacked, open up everything. And if you open up everything, if you dilate all these different blood vessels, you have this big drop in blood pressure, right? So in terms of anaphylactic shock and things like that, one of the quick responses that you have, people that have those types of allergic reactions, what do they usually carry with them? An EpiPen, right? An epinephrine pen. Epinephrine, we know, as a local factor, it's a hormone as well as a neurotransmitter technically, and we know the effects of that is it causes vasoconstriction, and it increases heart rate, and probably stroke volume and stuff like that. So if we have a big drop, EpiPen, right? Now all of a sudden we're going to get the response where we're going to be working to increase blood pressure instead. Okay? Okay, so that's vascular shock. And then finally, cardiogenic shock. So it's generated by a problem with the heart. Obviously, if the heart is not generating enough force, then that could drop your blood pressure. Okay. So, the remaining part of the chapter is really just a bunch of different figures showing you the different arteries and veins that we've been learning in lab. So it's a good reference in terms of those different blood vessels, but it's not necessarily something that we're going to be focusing on in a large part for the lecture exam itself. So every once in a while you may have a question having to do with blood vessel anatomy or something like that, in terms of more focusing on like the tunics and things like that. Every once in a while you may have a blood vessel question on the exam where it's like, oh, well, the main blood vessel that is going to like the forearm or something like that, or to the arm is going to be what? Is it going to be the subclavian artery? Is it going to be the femoral artery? Is it going to be the brachial artery, right? That's the main one of the arm, stuff like that. But more so, that's a lab type of question. So a lot of the rest of the chapter, if you're looking through your book, has to do with these figures that are really good for helping you to locate these different types of arteries and veins. Now, there is one other thing that I want to highlight before we finish up, and that was comparing our adult and our fetal circulation. Before that, Gabby, do you have a question?
Oh, yeah. This is just because I want to know. So
Yeah.
can adrenaline, you get scared, whatever,
Yeah.
can that cause like a shock to your like?
So adrenaline, epinephrine, same thing. And as we talked about, rather than cardiovascular shock, which is a big drop, it's going to go the other way. So it's going to make you hypertensive, right? It's going to increase. If you had normal blood pressure and then you do EpiPen, which sometimes you get curious about, you're like, okay, these EpiPens, they expire like other things. And just like other expiratories, you're like, really? Is it really bad? It's not that bad, right? It's okay, right? So every once in a while you wonder, like, what would that feel like the EpiPen? Don't do that. Don't do that. But it's a curious thing. If you have an EpiPen, you're like, I want to know. So in any case, if you had an EpiPen and you have normal blood pressure and you hit that, now you've introduced this thing that's going to increase blood pressure. So it's going to increase it. You're not going to feel too well, right? You might get heart palpitations and things like that. But likely with anything, right, it's going to cycle through. So it's going to increase it for a period of time, and then hormones and things like that get filtered out, and they get inactivated, and things are going to calm down. So it would go the other side. So its main response is to increase blood pressure. And so if you took it and you weren't low already, trying to normalize it, it's going to bring it up too high instead. Yeah. Okay, so the last topic here, which is a possible short answer question on the exam, is comparing fetal circulation with adult circulation. So this is something that we went over a bit in lab, but also we highlight in lecture, because it's a very unique system where we have these adaptations that allow the fetal circulation to interact with the placenta and do gas exchange, nutrient exchange, fluid exchange with that, but the adult doesn't have that, right? So what we see is that there are some adaptations, and you see them highlighted here, between the fetal circulation and the adult circulation. Of course, placenta counts as one too, right? Like we said, fetus hooks up to a placenta, the adult circulation does not. Now, in terms of these adaptations, as we were talking about it in lab, we have various things that will get blood to and from the placenta in the fetal circulation. So we, for instance, have two umbilical arteries coming off of those internal iliac arteries, two umbilical arteries that route this somewhat deoxygenated blood out to the placenta, right? We have one umbilical vein that then returns that blood from the placenta back to and towards the fetal heart. So going back this way, we have one umbilical vein. So we can see it like we saw it in the lab, right? We saw it on here, but then we also see it right here, right? So that's an umbilical vein going through the umbilicus, heading back towards there. Now, when we're returning blood to the heart, the largest vessel that does that in the body is going to be the inferior vena cava. So we've got to hook that umbilical vein into the inferior vena cava to get it back towards the heart. And that's the role of this vessel that we see right there, like we saw in lab. That is the ductus venosus. So we call it ductus venosus because it is essentially a duct, a small tube, that is returning blood back to the heart. So it's called venosus. So we have umbilical arteries, umbilical vein, right? There's two arteries, there's one vein, ductus venosus. We return that blood back to the heart. Because we've already exchanged oxygen and carbon dioxide, there are these shortcuts at the heart that circumvent or go around the lungs. Now, that doesn't mean that no blood goes to the lungs. The lungs would die off then, right? We still get some blood going to the lungs, but a large portion of it, a large volume of it, shortcuts, goes around that pulmonary circulation and goes straight into the systemic circulation so it can go out to the body instead. One of them is the foramen ovale, that hole that exists between the atria. And then ultimately, there's another one that is called the ductus arteriosus that connects the pulmonary trunk to the arch of the aorta, right? So those are modifications of the heart or near the heart that are also in the fetal circulation and not in the adult. So the ductus arteriosus, because that's going away from the heart, becomes the ligamentum arteriosum. That's what we see on the adult heart model. That little white fibrous connection between the pulmonary trunk and the arch of the aorta. It's on the list of things we have to know, so make sure if you don't remember that you check it out. So the ductus arteriosus becomes the ligamentum arteriosum. Foramen ovale becomes the fossa ovalis, and we can see that on the adult heart model as well, right? It looks like a thumb depression instead. It's a fossa, the depression instead of a hole. Ductus venosus becomes the ligamentum venosum. Again, a fibrous connection instead. Umbilical vein becomes the ligamentum teres. So the rest of this that we see down here is ligamentum teres. And then the umbilical arteries become the median umbilical ligaments. So there are still connections that remain, but they're not functional. There's no blood going inside of these fibrous connections, but there's still kind of dried up remnants that are there. I was telling some people, I think in the second lab or something like that, right? So you can actually feel, if you so choose, you can actually feel those median umbilical ligaments. So if you take your fingers and you put it in your umbilicus, right, put them in essence, in your belly button, and you move it around, like significantly, you can actually feel a pull on the inside because you have those median umbilical ligaments going down and connecting to those internal iliac arteries. I mean, you don't want to be like, or anything, but you can actually feel a pull in there because there's still a connection down into there. All right, so the point is that we need to know these adaptations between fetal and adult circulation. You need to know what it's called in the fetus and what it turns into in the adult. Yeah.
So if we had the short answer question on the exam
Yep.
about this, would we just put like foramen ovale becomes ossovallus and ductus arteriosus becomes ligamentum arteriosum?
Yeah. Yeah, I mean, I would, for the fetal structure, because those are functional structures, I would maybe put just a short sentence of what it does, right? Foramen ovale connects to two atria, something like that, and then, like you said, turns into the fossa ovalis. Yeah, that would be fine. And in a lot of cases, if we're asking you to do comparisons or something like that, like different types of blood cells, put the name of it, put the function of it right next to it. You don't even have to write it into like paragraph format or anything like that. You can just do your list, okay? If you're doing the blood flow, so if you're asking about, if we've asked you to trace a red blood cell as it passes from the systemic circuit until it reaches the lungs, that's basically the right side, right? Deoxygenated, stuff like that. You could just list, right? Like the left atrium and then put an arrow or a dash to left AV valve or left atrioventricular valve to, you can write to if you want, to the right ventricle, through the pulmonary valve, through the pulmonary trunk, pulmonary arteries, to the lungs. Done. You don't have to necessarily even do descriptions there. This to that, to that, to that, just like we had it written out before. Okay? So it's okay for you to be kind of reductionist and just list, as long as you have enough information there, just list what the question asks for. Right? Here's the structure, here's the function, whatever. Different types of blood vessels, here's the name of the vessel, this is its structure, this is why it has that structure. Okay? I can tell you, on that question, if you get that question where it's asking you to compare different blood vessels, like list 5 different types of blood vessels, and it asks you to discuss their structure and function. What's unique about their anatomy and their physiology? Make sure you put, it doesn't have to be anything big, but make sure you address both those things. So that the capillaries have only a single layer to their wall, that the arterioles have a small diameter and they have a really thick, smooth muscle layer in the middle or something like that. Make sure that you, and then, of course, like we said, they are reducing the pressure for the capillaries, or in the capillaries, it allows for exchanges with the tissues. You're linking the structure to the function. Sometimes people will just name the vessel and what it does and then forget that it asked about some aspect of its structure as well. Okay, so make sure you're reading the question and answering all the questions. Review your answer. Do I have all 5 things in there? Did I answer all the parts of the question? Okay? Okay, yeah.
For the ductus venosus, and it turns into the ligamentum venosum for
Yeah.
the adults, what is the function for the adult one?
Nothing.
Nothing. It's just
So, yeah, so when we're talking about the function, if you are in the fetus, all of these things have a function. Routing blood to and from the placenta, back into the fetal circulation, circumventing the pulmonary circulation, things like that, getting it out to the body faster. But then in the adults, it's just like leftovers. Yeah, so no function. You don't have to worry about that. But why was it there originally? Okay? Yeah.
What does the ductus venosus bypass?
So, good question. So the ductus venosus, its role is to connect the umbilical vein to the inferior vena cava. So it's not necessarily that it's bypassing, going around anything. It's this extra little extension that's going to get that umbilical vein that's coming out here from the placenta. It's going to get it hooked into the inferior vena cava.
Okay.
So that's it. So you can just say that it's connecting those two vessels together. That's all right for a function. Yeah.
On the ductus arteriosus.
Yeah.
Is that connecting the aorta to the
Pulmonary trunk.
pulmonary trunk?
Yep.
Okay, that's what I thought.
Yep.
I was putting IVC on there, and I'm like, that's not right.
Mm-hmm. Mm-hmm. Okay. So those are our comparisons that we want to make between our fetal circulation and our adult circulation. Okay? All right, so that's the last topic for the blood vessel chapter, hemodynamics and stuff like that. Questions? Really opening up in terms of questions for anything. Blood vessels, blood itself, which it's been a little while, right? That was our first chapter in our first weeks or whatever. And then heart.
So if we were to be given this question, it was like one of the bolded objectives too.
Mm-hmm.
Can we also say just like the resistance is different and the blood carrying capacity is different between?
You're not talking fetal circulation.
It's like 28. It says know the structural and functional differences between fetal and adult circulation. I can say placenta, resistance, and the different blood carrying capacities between both of them. Like,
I wouldn't worry anything about resistance or anything like that in terms of hemodynamics. Just straight up kind of like what each of those things are doing. Placenta is all about, exchanging with a maternal blood pool, nutrients, oxygen, things like that. That would be fine enough to talk about the placenta and its role. And then all the rest of these connections, if you just discuss, okay, why do we have it there? What's it connecting from here to here? Why is it doing that? Like we said, getting the umbilical vein into the inferior vena cava. That's okay to describe why it's there. Yeah. All right, so I know there are a few questions out there. Again, still about our different types of cardiac muscle cells. So we can talk a little bit about that again. We can review that again. There were some questions out there maybe about blood vessels as well, EKG, things like that. So for sure we can discuss that as well and anything else that you are interested in. So if we're talking about autorhythmic, right? And with these ones, you're not going to be required to draw this out, but you are going to be describing the similarities or differences between the two. So I'm doing like the graph thing, but if you wanted to, you could create a table to help you organize your thoughts as well, right? So you could go, okay, autorhythmic and contractile. And you can compare them in various ways. You could be like, okay, so what is the ion for depolarization? What's the ion for repolarization? Does it have a resting potential? Does it, let's see, have that weird plateau thing? EAU, like that. You could say, for the cells, right, two different types of cells, like, what's the frequency of them? Like, are they the 99% or are they the 1%? You could say where they're located, right? Are they located in the myocardium for the contractile cells? Are they located in the conduction system, like the SA node, the AV node, and stuff like that for the autorhythmic cells, right? If you wanted to get into numbers, you could get into some of those numbers, like where they start and where they stop. They both go up to about 0 millivolts, but like our contractile cells are down at about negative 90 millivolts or so, And our autorhythmic cells are vacillating between like negative 60 millivolts and then up to the 0 or so. So you could say the baseline for them, the lowest point, is a little bit different. It's okay when you're doing your comparisons if your answer is the same, okay? The ion responsible for repolarization in these is potassium in both cases. That's okay, right? We just want to be able to understand how this one works, how that one works comparatively. Okay? So our autorhythmic cells, we're going to be having this, right, where it is rising slowly, then quickly, and then back down, and then rising slowly, and then rising quickly, and back down like that. So this is the one where we don't have our resting potential, really. And this slow part of the curve, right about here or so, right, this raising part that's here, this is all depolarization. This part is due to sodium ions. This part is due to entering calcium ions. This is both in. The repolarization is the declining part of the curve, and that is due to potassium ions that we see there. In the contractile cells, we do have this kind of resting potential like this. Then we have a very fast depolarization, this wacky and elongated plateau before we go back down to resting potential again. And this is more like negative 90 millivolts. This fast depolarization is due to sodium ions coming in. This plateau is due to calcium ions continuing to maintain it in a depolarized state. So voltage-gated calcium channels opening up there. And then the repolarization is the same. I don't know, that kind of looks like a T, but it's a plus. And that's potassium exiting the cell as well. Okay? This is on the order of like 5 to 10 milliseconds in terms of the duration of it. This is on the order of like 250 to 300 milliseconds in the duration of it. So you can compare that as well, right? How long it takes. The duration of those action potentials is vastly different. So I'm listing a whole bunch of different things. You need to be able to list 5, right? Because it's a 5-point question. We want to be able to highlight, okay, what are the 5 things that we can see in that answer? Okay? Questions on that?
Does it say negative 90 on the contract?
Yes, sorry.
And is that where the, like the baseline?
Yeah, like where that baseline, where the resting potential is, about that.
So the movables go, do
What's
they
that?
go, do they go up?
Do what go up? Those, yes, I'm sorry. So this is about 0 millivolts here. And this is about, again, about 0 millivolts there. It overshoots it a little bit, so it goes up to like maybe plus 10 millivolts or something like that before it returns down. Yeah. Yeah.
So on the autorhythmic, they don't have a resting potential, but the action potential is that the pacemaker cells, the slow leaky sodium ions.
Yeah. So that pacemaker potential is kind of what starts that whole rhythm like that. So it does not have a resting potential. Each one of these spikes that we see here is an action potential that starts with sodium leaking into the cell as part of a pacemaker type of thing. Like, what's the rhythm going to be? And again, like we see even with the EKG, which is a separate topic, the split between these, the spacing of these can change, right? We know that heart rate changes. It's changing right now. If you take a deep breath in and take a deep breath out, right, that'll slow things down. The heart rate will naturally slow down as well, okay, as well as the breathing rate. If you start thinking about something that's stressing you out, now it starts going as well. So how closely these are spaced, that can vary, but the overall pattern of it that we're seeing between these two is going to be the same. Okay? Okay. So let's take a look at ECG as well. Now remember that the ECG, which is what we were doing in lab today, so that'll help us to review it. It is electrical activity, so it's still going to be time on the x-axis, in seconds in this case, and millivolts on the y-axis, okay? So this is where we have boom, P wave, boom, 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 QRS wave, and then boom, T wave, right? That's one cardiac cycle. So for those 3 waves, we need to be able to label them. What's the name of each one? P, Q, R, S, and T. We need to know what's going on during each of those. So for instance, P wave is equal to atrial depolarization. Okay? That's when the atria are first getting their signal to contract. So in this period following that P wave is when those atria are going to contract and eject their blood. Okay? So this is the electrical activity. This is the electrical activity that we're seeing in those waves, right? And it's followed by the mechanical activity. During the QRS wave, we have atrial repolarization now, and now the ventricles are getting their signals, ventricular, boink. So they are going to be doing their depolarization during the QRS wave. So 2 different things going on. That's why it's really big. T wave is equal to ventricular repolarization, which is going to be followed by their relaxation and filling. Okay? So identifying the 3 waves with the appropriate letters and being able to describe what's going on during each of those waves is the critical aspect about that. And then wherever it says depolarization, right afterwards, that's when contraction is going to happen, or systole. Wherever it says repolarization for that chamber, right after that is where our relaxation and filling, or diastole, is going to occur. Okay? And remember, this is for the heart overall. So, even though it's electrical activity on a graph like that through time, we're saying each one of these spikes here is just, whoa, something's going on in the heart. Okay? So it's an electrical activity of the heart. That charged particles are moving, much like even with skeletal muscle or with neurons and stuff like that. So this is atrial activity before they're going to start contracting. This is ventricular activity plus atrial activity. And then this is, like we said, ventricular repolarization that we see there. Okay? So again, totally fine if you want to draw that out on scratch paper or something like that. When you get into the exam, you're like, oh, I want to label all this stuff, and this is what it is. But on the exam as a short answer question, you'd just be describing that. What are the 3 waves of the EKG? P wave, QRS wave, T wave. What's happening during each? Atrial depolarization, et cetera, et cetera. Okay? Now, it could be, whenever we're talking about these topics and we say, oh, that could be a short answer question, that doesn't exclude it from being a multiple choice question as well, right? So you could have a multiple choice question where it says, during the T wave on an ECG, what's happening? Atrial depolarization, atrial repolarization, right? Ventricular depolarization, ventricular repolarization. Oh, that's the one. Okay? It's just the multiple choice questions usually are very detailed, asking about just one thing at a time, right? Whereas the short answer questions are more of like, what's a concept? How does this thing work? Compare these various things in different ways, etc. Okay. What else? Yeah.
On the blood vessel section, I think it's objective 27 says, clarify changes to cardiovascular parameters and blood flow to organs, and it lists a bunch of organs during exercise. Can you clarify?
Mm-hmm. So, and it lists organs like what? Like kidneys?
Kidneys, and let me look it up. It had a bunch of them listed, and according to the book, the blood flow would decrease, I believe, in the kidneys, and I can't remember the other one, but we never really went over it, so I was a little confused.
Yeah, so good points for that to review. Number one of the things that we keep highlighting is that, like with the textbook, so you should be using the textbook as a reference for sure, right? Check out those tables and those figures where it goes through these things in a different way so you can kind of reinforce that knowledge that we're talking about here in lecture, okay? read through sections as well, because they have a different way of describing those same topics that may click with you better than when we were talking about it here in lecture, okay? But if you get to a section of the chapter and you're like, gosh, I don't think, if I look at my PowerPoint handout, which is my other major reference for reviewing what we covered in lecture, and we didn't address that topic at all, then it's likely not going to be on the exam, right? So the lectures are helping us to focus on the major topics that we're going to be assessed on, right? Because the chapters are bigger and they cover a lot of different things, and sometimes, right, if you're going to go into whatever, if you're going to be a cardiovascular surgeon, then you're going to need to know quite a bit more on certain topics, etc. So they're a bit more broad that way. So use that as a guideline. The more time we spend on something in lecture, that means the more questions you're going to have on it. If we didn't bother to mention it, or somehow we didn't, and you're looking through, like there was a question in there talking about the hepatic portal vein. So hepatic portal vein, definitely a vein. Hepatic means liver, right? So there's something going on with that. We didn't mention it much here, okay? So it's likely not going to be on the exam. Every semester it kind of takes a little bit of a different direction in terms of topics that we may or may not address. We always hit the big ones, but sometimes we'll go off on other forays. Hepatic portal vein probably won't be on there. It's important. What it does is it's, interestingly enough, taking nutrient-laden blood from the intestines where it got absorbed, taking it up to the liver for processing, storage of glucose and things like that. So it counts as a vein because it's on its way ultimately back towards the heart, but it's actually porting or carrying these nutrients to the liver for processing instead, okay? So guidelines like that. But the other specific question was asking about blood routing to different organs as to whether you're exercising or not. So won't be as critical in terms of the exam, but we want to highlight the fact that the body is all about adapting to the needs at the time. The needs of the whole body. Is it warm or cold in this room? And so I got to warm or cool the whole body to maintain homeostasis. But also, we have needs in different areas of the body at different times as well, okay? So if you are exercising certain muscles, right? If you are bench pressing or something like that at the gym, then, wow, we need to route blood out here because those muscles are working hard. I'm sitting down while I'm doing that, so my lower limb muscles don't need that. So we're going to dilate certain blood vessels, constrict other ones in order to route this blood to the places where we need it to be. So that overall concept is important, but in terms of like looking at specific organs and things like that.
Yeah, this skeletal muscle, so it says, describe how the following cardiovascular parameters change during exercise, and then it lists heart rate, stroke volume, venous return, cardiac output, blood pressure, total peripheral resistance, and then it says how it affects the blood flow to skeletal muscles, brain, skin, digestive system, and kidneys. And I just noticed in the book, I think it said kidneys and maybe digestive system. I can't remember.
Are
that
reduced?
it decreases,
Yeah.
but then everywhere else it increases. So
Right.
I'm just confused.
So that gives you an actual, it just allows you to think about what we're learning, like where are the needs, right? So I gotta always have blood going to the brain. If I'm exercising, gonna have to go to skeletal muscles and things like that. And in order to do that, and if we're gonna increase blood pressure and blood flow, we know that there's 3 different things that contribute to that. The blood contributes, the heart can contribute, and the blood vessels contribute. So if we need to increase blood pressure or something, we're going to increase heart rate. We're going to increase stroke volume, right? So the heart can have those types of responses. The blood vessels can dilate or constrict. If we need blood pressure to go up, we can constrict them. If we need more blood flow, we can dilate them. So it just kind of gets you thinking about how the body is using these individual mechanisms that we've been learning about to meet the needs of the body at the time, right? So the kidneys are about filtering the blood. They need to do that all the time, but we can prioritize that or not at certain times. Same thing with the digestive system. Rest and digest, parasympathetic. Later on, after you've eaten and you're resting and stuff like that, and the muscles don't need all that blood flow, let's route it to the stomach and the intestine and stuff like that to be able to pull those nutrients in and get them into the circulation. So again, we can route the blood where we need to, prioritize it to certain places. Yeah. That also reminds me, I sent out an email about this. this not only relates to possible short answer question, but it also needed a clarification in terms of the colors of the arrows that we see here for local factors that affect blood vessel diameter and whether it's dilation or constriction. Okay? So it is correct that the green arrows that we see there are going to be dilation, right? So green light, go. Red light, stop. And it's not go and stop, but it's like increased blood flow, right? As opposed to decreased blood flow. So I think the original slide provided by like the book publisher or something like that had those reversed. But if you think about it, just like we were going through in that exercise, if, for instance, like in the upper left-hand corner, you see oxygen levels in a tissue drop, well, do we need more blood flow there or do we need less blood flow there? More, because all of those cells, like skeletal muscle cells, they're like, I need more oxygen to make more ATP, right? I'm going hypoxic. So we need to dilate those blood vessels in order to give them more fresh loads of oxygen, right? So these are local, like neighborhood environmental factors that can affect and determine what those blood vessels are going to do. It says, hey, I'm letting you know I need some more, or I don't need as much as well. So higher levels of carbon dioxide, that means that that tissue is very active as well. And we need to kind of flush that out of there, right? So higher concentrations of carbon dioxide in the surrounding environment can lead to vasodilation. A drop in pH, so hydrogen ion concentration or a drop in pH, that also indicates an active tissue like lactic acid and things like that. Increase in potassium levels, again, with that one, what's happening? Repolarization, we open up those channels, potassium is flowing out of the cell. That means there's a lot of action potentials in those cells, whether they're skeletal muscle cells or whatever. So if there's a lot of potassium showing up in the external environment, that's another indicator, ooh, that tissue is very active, it needs more blood flow. Okay? There's other things like temperature comes into play here as well. So if you put a cold compress on something, that leads to vasoconstriction, right? If you have sprained an ankle or something and you don't want that swelling, you want to reduce the swelling, cold as a temperature causes vasoconstriction. Meanwhile, heat causes vasodilation instead. So that can affect that. But think about it in terms of an active tissue. Again, we keep using the example of skeletal muscles. Skeletal muscles, when they're active, they're generating heat. So when they generate heat, that's a stimulus to these blood vessels. It says, oh, you're working hard, we better give you more blood flow as well. Okay? Okay. And then we see a few other things in there too. Some of the hormones that we indicated, that can be vasoconstrictors or vasodilators. There's our epinephrine again, that comes into play. So a variety of different things that can affect the blood vessel diameter. Okay? So if you got this as a short answer question, again, you're not going to be expected to draw arrows or anything like that. Asking for probably 5 examples. And what you need to say is, if that factor goes up or goes down, how does the blood vessel respond? Okay? So oxygen, that was our first example. If oxygen levels in this example, if oxygen levels go down, the blood vessel is going to dilate. But the opposite's true as well. So if there's high oxygen content in that tissue, it's not working very hard, right? It's banking a lot of that oxygen rather than using it. So in that case, we may do vasoconstriction and kind of route it through there a little bit. It doesn't need quite as much blood, and we can get it through there faster, right? So you just need to say for each of those factors or any factor that you give, if that factor you can choose, that factor went up or down. Is it heat or is it cold? And then how does the blood vessel respond? Does it dilate? Does it constrict? Okay? And that relates to, like we said, blood flow to that area. Okay? Okay. What else? Yeah.
So as a general rule, basically, dilation means blood pressure lowers and restrict or
And blood flow increases. Yeah. So it's always it's an odd thing in terms of hemodynamics or hydrodynamics. So again, we keep thinking of a hose. If you kink a hose somewhere, that affects things upstream and downstream, right? So if we kink the hose right in the middle, downstream, our flow and pressure significantly affected, right? We have a reduction in flow that we see there. But upstream, we have a reduction in flow and we have an increase in pressure because all of the fluid is arriving there and it doesn't have any place to go. So when we constrict or dilate, that also comes into play. Like, where are we talking about? Generally we're talking about downstream, like what we're supplying, if we're thinking about arterioles and stuff like that. But it does have that effect. If we cinch down a blood vessel, that's also going to increase blood pressure, right? We think about that, like we said, stress response, epinephrine, stuff like that, cinching down these blood vessels and increasing blood pressure. So there's kind of this relationship, like where are we talking about, downstream or upstream, if you will, from where that constriction is, and when we're talking about flow and pressure, etc. Generally, like we said, we're talking about arteries and arterioles supplying these things. But we know that veins have all three layers as well. So that is another topic, right, where we're saying, okay, what are those three layers? There's tunica externa, tunica media, tunica interna. What are the different tissues that make those up? And how is that similar or different in a vein compared to an artery, compared to an arterial, compared to a capillary, right? Capillary just has that single layer, whereas all of our other larger blood vessels have all three layers. An endothelial layer, that's our lining on the inside, that prevents, hopefully, that blood clotting cascade and stuff like that. A middle layer, which is smooth muscle, which allows us to vasoconstrict and vasodilate. And then, generally, a connective tissue outer layer. Connective tissue fibers like collagen fibers, as well as elastin fibers and things like that that are in our outermost layer like that. And then, if we have a capillary, that's the extreme of anatomical and physiological linking, right? Super thin wall. We still have a lining because we don't want clots there, but we have missing layers of the wall, right? We have a super thin layer that facilitates that exchange. That makes it easy for things to get across the wall. It's very difficult for things to get across three layers because they'd have to march across all three layers and they're very densely packed and stuff like that. And by the time we started doing that, the blood would be out of there already, right? So, other things, blood clotting cascade as well, right? That was one too. You're comparing our extrinsic and intrinsic pathways. You know that there's two of them, okay? Why is it called extrinsic and intrinsic? Outside the vessel, inside the vessel. And then what initiates it, right? Outside the vessel, we're talking about things like tissue factors, etc., things that it's come into contact with that it normally doesn't see, but in the surrounding tissues, as opposed to on the inside of the vessel, we've broken through those inner layers, and now we're seeing collagen fibers, etc., of the vessel wall. Between the two, the intrinsic is much slower because it uses the full pathway of all the different clotting factors, whereas extrinsic is quicker because it only uses a subset, a shorter number of them, kind of jumps over some of the steps. But they both result in the final result is the activation of fibrinogen, one of our plasma proteins, into fibrin. Fibrin layers forms fibers onto that platelet plug, strengthening it, catching other blood cells and things like that, and forming a very solid clot. Okay? And then when we need to get rid of that clot, plasmin, which as activated from plasminogen, is what is eating away at those fibers, right? So it's lysing and eating away at those fibers and dissolving the clot. That's the primary plasma protein that is working to get rid of clots instead. Other things. Blood typing. Don't forget about that. Okay? So, of our different blood types, what does it mean in terms of type A, type B, type O, etc.? Yeah?
Can you go through when it's O positive versus O negative, which one is the
Universal donor?
Yeah.
Yeah. So when we're talking about that, right, when we're talking about our blood cells, the different types that we have indicate what antigens are attached to the cells, okay? So if we had an example like this, And there are various, again, check the figures. These are two different figures trying to show that in terms of antigens and antibodies floating around. I kind of, like the other one, this one, a little bit better, but either one's fine. But what you need to look at is that with the blood cells, we see type AB, then B, then A, and then O in this example. You can see that type AB blood cells have both the A and the B antigen on them, okay? So the easiest thing about the blood groups and the blood types is that whatever you see listed here, that means it has that type of antigen on it, okay? Now type O, like I said, I think of it as like type 0 instead of O because it has neither one, all right? But all the other ones, whatever letter you see there, that's the antigen it has attached to the cells. Now the other thing that you have to think about with that is, as Brita was saying, the positive and negative. So this has to do with an A and a B antigen. Do you have them both? Do you have this one? Do you have that one? Do you have neither? Okay? But the positive or the negative has to do with an additional antigen, the Rh antigen. So if your blood is tagged as positive, that means you have that additional antigen on top of your cells to be identified. Okay? So AB positive has the A antigen, the B antigen, and the Rh antigen. AB negative has the A antigen, the B antigen, that's the AB. Negative doesn't have that antigen, that Rh antigen. It does not have that. So that's the negative aspect of it. So each of these blood types, the other subset is, do you have that Rh antigen, positive or not, negative? Okay? The last part about this is not only do we need to explain what antigens are attached to the cells, but what antibodies are floating around in the plasma trying to protect against foreign cells, cells that do not look like yours. So the antibodies, number one, are floating around. That's one of our plasma proteins. Remember in the blood type question, if we're asking you about name 5 things that we find in the plasma, ions, respiratory gases, water, nutrients, antibodies are one of 3 different plasma proteins that we find in there. Knowing what each one of those plasma proteins does is important too, right? So if we say albumin as a plasma protein has a function of, and then we have a multiple choice question where it's like determines blood type or causes clots or helps with osmotic pressure, things like that, right? You can tag and meet up with the function and that particular plasma protein. Okay, so antibodies are plasma proteins, also known as immunoglobulins floating around. The antibodies are for tagging cells that do not look like yours. You don't want those sticking to your own cells, right? It would form clumps like we saw in the blood typing card. You don't want that agglutination happening. You don't want those clumps like you see in the lower right-hand corner there. So those antibodies are matching up with foreign antigens. So if you have type A blood, you're going to have antibodies against the B antigen floating around, okay? So it does not match with your own antigens. It would match with any cell that had an antigen that is unlike yours, right? So your type A cells have the A antigen. That means any cell that has a B antigen is not yours, right? So we have an anti-B antibody floating around looking for any cell that has a B antigen. It's going to stick to it. It has a complementary shape. It's going to stick to it and agglutinate with it. Grab it and kind of hold on to it so our macrophages can come eat it. Okay? So for the different blood types, you need to know what does it mean in terms of the antigen and what does it mean for the antibody. And the antigen is on the cell, attached to the cell. The antibody is floating around in the plasma, unattached. Okay? Yeah?
So for the positive and negative, the positive can take negative, but negative can't take positive.
Yes. So thanks for bringing that back around. So in terms of like O negative blood, that means it does not have the A antigen, does not have the B antigen, nor does it have the Rh factor. So they don't have any of those things sticking out to be identified, which means that it very easily can be given to somebody else and it does not provoke or basically invoke an immune response, right? Because there's nothing for it to identify and tag onto. It's kind of going under the radar. So for an RH factor, if it's negative blood, that means it's one less thing to be identified or flagged by the immune system. Okay? So O negative is going to be that universal donor. If you have somebody with negative blood, you can't give them positive because they have that extra flag that the immune system is going to see is different from their own cells and glom onto it, grab onto it with antibodies. Yeah. Okay. What else? You got about 5 minutes or so. Ish. 5. 3. Because that clock's always fine. Yeah.
I covered this a little bit, but I just wanted to clarify. So on the blood vessels, I think it's the third objective says describe the structure and function of the endothelium. My question was in regards to the tunica interna or the capillaries or both. You kind of talked about the function already, but this question is vague. It's not saying if we're talking specifically about the tunica interna or the capillaries, or do we want to talk about the structure and the function of the endothelium in both things?
So as a layer, when we're talking about the different layers that make up blood vessels, the endothelium, the innermost layer, which is a type of epithelium, is about creating this smooth, non-reactive surface for the blood to scoot by and not react with. So that's still the case with our capillaries. It's just with the capillaries, we get rid of the tunica media and we get rid of the tunica externa, so we only have that. And that also, the thinness of the wall overall there, makes it easier for things to exchange across. So, yeah. It's the fact that we only have that layer which makes those blood vessels quite well adapted to exchange. So as you're looking through things to see if we have any final questions, in the remaining days as we're leading up to Saturday, okay, it is my recommendation, number one, that you're going out, you're checking all of your different resources. Your PowerPoint handout, that's one of your biggest resources. Your Panopto videos, right, that are searchable. You can type a term in there, and you're like, albumin, where is that in the blood lecture? And it'll tag every slide, take you right to it, okay? Your vocabulary list and your objective sheet. Notably, those ones in bold, okay? You want to make sure that you're reviewing those a lot. And when you're reviewing, don't just like read it and you're like, yep, I read it 3 times, I got it. Quiz yourself on it. Create, flashcards or something like that where you can actually demonstrate to yourself how you're progressing in terms of your learning of it as well. And if you want to take that one step up, it's one thing to do your own flashcards. You're like, okay, yeah, I'm feeling pretty good. Whoa, those not so good. Put yourself out there. Give those flashcards to somebody else or hand somebody else your objective sheet with your written answers and say, ask me any of those ones in bold. They don't have to know anatomy and physiology. They can see all the words and be listening to your answer like you didn't talk about this tunica media thing, right? Put yourself in that situation where you're a little bit uncomfortable because you're actually being tested. Because that's more similar to what it's going to be like on the actual exam, right? If you're just sitting there, you're like, this is good. It's like kind of reading over the driver's test and actually doing the driver's test. Nobody does a driver's test without practicing behind the wheel. So in the coming days, make yourself uncomfortable until you're comfortable. Put yourself in those situations where you're actually testing yourself. So you can see, look, I'm good, I'm confident in this. Every one of these questions I got, if I get those, I'm going to nail it. That's good. And then it also allows you to see, these couple of questions I didn't get. I don't have it yet, so now I'm going to focus my study efforts on that at this time. So it makes your studying in those final days more effective as well. It's not like everything, like where do I need to prioritize my time coming up? Okay? Okay. I got office hours today after lab. I'm available by email as well if you need to, if we need to do something by Google Meet, like by appointment or something like that, we can do that as well. I should be on email Saturday, probably in the middle of the day, late afternoon or something like that for questions as well. So feel free to send me emails about things that you're still struggling with, that you need help with, etc. if we're not together. Okay? Okay. So, morning lab people, you are free to go. And later lab people, we will see you next door for cardiovascular physiology.